So it's really my great pleasure to introduce our second speaker, Patrick Wincker from the Genoscope in France. So Patrick got his PhD from Marseille University, and then he moved to Brazil for 6-7 years, where he worked in parasitology, and more particularly on trypanosis and leishmania. And he came back to France and then was appointed in 1997 at Genoscope, first as the head of a sequencing group, and now he's the head of the sequencing platform and coordinates all the eukaryotic genomics projects. The Genoscope has played a major role, and I know many of you know about it, in the human genome sequencing, but since then have been working on a lot of other genomes, and I've counted more than 120 organisms that were sequenced at the Genoscope, and from all different taxa and different worlds. And more recently, Patrick has coordinated projects in the plant genome, so grape wine, banana, coffee, but as well fungi and protists. And since 2009, he's the scientific coordinator of this great project called Tara Oceans, and he's coordinating all the genomic activities there and the sequencing. And he will tell us about, it's a fascinating expedition. So this is a boat going around the world and collecting water and measuring all kinds of characteristics and sequencing what is found in these samples all over the world. It has started in 2010, and I think Patrick had the chance to go once on this boat. I don't know if this picture was taken from the boat, but you can imagine it was shaky from time to time. And the goal of the project is to explore the diversity of the plankton and understand actually the impact of climate change on the ecosystem in the ocean. So very looking forward to listen to your talk, Patrick.
Thank you, Catherine. So first of all, I would like to thank the organizers for the invitation. So my talk will be about the challenges facing the use of metagenomics analyses on communities that are dominated by eukaryotes rather than prokaryotes. And I will discuss that mostly in the frame of the Tara Oceans project, where the environment that is studied is plankton from the photic zone. So if you look at a generic environmental genomics project, its main purpose is to describe communities and their functions, so the biotic components of the communities, and correlate them with physicochemical characters, with the physicochemical context, and explain how the communities is interacting, how the physicochemical context is changing due to the activities of the community. So this is done generally in most projects that you know from metagenomics. These environmental projects are done using prokaryotes and viral communities essentially. This is due to technical hurdles basically, but also to the thinking from many people that most communities in the microbial world are really dominated by bacteria and sometimes archaea, and where eukaryotes play a rather minor role. This is not always the case, however, and we know that in many environments, and this is particularly true in plankton, in marine plankton, as eukaryote components, mostly microbial, but I will also talk a little bit about small metazoans. They are not only understudied, but they are really responsible for a great part of the biogeochemical activity. Many of you probably doesn't know how complex is the plankton community, so this simple slide is just showing you what is in 1 liter of seawater. So this is a mean composition. Of course, it is extremely variable, but we can discuss it in containing a large number of organisms. The most highly present organisms are the smallest one, in this case viruses. So when you take 1 liter of seawater, you have billions of viral particles everywhere. You have also close to billion prokaryotic cells and up to hundreds of millions of eukaryotic cells, which are in fact relatively abundant, but also far bigger than most of the prokaryotes. And you have far less metazoans, small metazoans in plankton, but still a fairly large number of them. So in many cases, you can see that prokaryotes are indeed dominating in terms of cell number, but in most, in fact in many plankton communities, eukaryotes are in fact dominating in terms of biomass because of their larger cell size and larger organism size. This is shown here in this graph, which shows the biodiversity estimated. We have taken this graph from different studies, compared with the cell or the body size, so the size of the organism or the particle. We have an extremely large number, which is in fact relatively unknown today, of diverse viruses in the sea. But we have a finite component in prokaryotes, which is not completely known, of course, but which is estimated up to something like around 1 million different species. And eukaryotes are probably as numerous in terms of species number, or maybe even more numerous in plankton, particularly the unicellular component, which in size is overlapping the largest bacteria and is also overlapping some of the smallest metazoans. So they run from less than 1 micron up to cells that can be up to 100, 200 microns in size and sometimes more. So this is resumed here. You have a classical tree of the eukaryotes that is shown here. You can see, or maybe you cannot see, but your favorite organisms like land plants are in one line just right here. Animals and fungi are 2 lines here. And the eukaryotes are a fairly large diversity of organisms that are really, really understudied today. Here you have different groups, and all the red arrows are pointing to phyla that are found in marine plankton. So you can see that marine plankton contain a very large number of different type of phyla of the eukaryote, amebia, straminopiles, alveolates. All of them are represented, and many of them are represented not only by photosynthetic groups, but also by heterotrophic groups, parasites, symbiotic organisms, and so on. So there is a very huge complexity of single-cell eukaryotes in the plankton that is today mostly undescribed. In many cases, you don't have any genomic information for these groups, and even in the cases where some phyla have some genomic information, it is rarely from marine organisms. So let's discuss a bit the objective of the expeditions that we have created in order to study marine plankton in a relatively different way from what has been done until today. So this expedition is called Tara Oceans. Tara is a boat. It's a small boat that has been used across the world oceans to create this collection of marine plankton samples. So you have here the travel of the boat during the 2.5 first year, 2009 to 2012, across the different oceans. This expedition, this part of the expedition is completely finished now. It has go back to its home port in the end of 2012, and there has been a second part dedicated to the Arctic Ocean that just finished in December. So we now have all the samples from these different expeditions, the 2 different expeditions, and as you can see here, they have crossed most of the main biomes that can be seen in the oceans, the North and South Atlantic, the Indian Ocean, Mediterranean Sea, a great part of the Pacific Ocean, the Antarctic, and now also the Arctic. So we have collections of samples. I will show you how they are collected and how they are analyzed from these different locations that are ready now to be studied, and part of them have been started to be studied. What is the idea behind the study of the plankton? So many of you are not working with plankton, but the importance of plankton can be seen in very different aspects. One of it is that, of course, the plankton is the first step in the trophic system of the ocean, so it's really where photosynthetic activity starts and where all the further steps are initiated. Plankton, of course, is sequestering a large amount of CO2, and this has many implications in the current studies of climate change. Many people doesn't know, but about 1/2 of the oxygen that is produced on Earth is produced by land plants, and the other half is indeed produced by photosynthetic organisms in the ocean. So it's a very important ecosystem in many aspects. So this is a boat. This is Tara, a relatively small boat, a schooner that has been used all across the expedition. This boat has been constructed, in fact, to go to polar environment. So it's a relatively resistant boat that can be that has been used really in many different conditions, not only in beautiful weather as in this picture. So we have done 3 years of sampling, and in total 200 sites, approximately 200 sites were sampled. And together with these 200 sites, there has been continuous data acquisition, which means that during the travel of the boat, there is a constant amount of seawater that is analyzed inside the boat, and there are some imaging and some physical characterization of seawater, even if the boat is not stopped in a particular station. The idea is to sample all microscopic life, so from viruses to fish larvae. This is something really different from many other expeditions, like for example the Sorcerer expedition of Craig Venter, which was mostly dedicated to bacteria. And the idea here is really to describe the plankton as an ecosystem very globally. But we sample only the photic zone ecosystems. And there are planktons at different depths in the oceans, and the aim of the Tara Oceans project is really to look at the photic zone, mostly at the subsurface and at depths up to 50 to 100 meters, and not more than this. And it implies a lot of multidisciplinary approaches. So the consortium, I think it's my next, no, it will be another slide, but I will show the consortia working on this data, which is really not only composed of specialists in genomics, but of many aspects of oceanography. We described the approach in a kind of white paper 2 years ago in PLOS. So this is the title of the paper. Our idea is really to try to have a holistic vision of plankton using different methodologies. So there is a component in high throughput sequencing. This is what I will most talk about today. But there is also a high throughput imaging in order to record morphology of the plankton and try to correlate it with the genetic data that we will obtain. And of course, there is a lot of physicochemical measurements that we'll use later on to try to establish correlation between plankton population and these different parameters. So this is a consortium. It is directed by Eric Carcenti from EMBL, and it includes many scientists. Some of them are specialists of diverse type of plankton, like viruses, large viruses, prokaryotes, eukaryotes, and so on. It includes specialists in imaging, specialists in genomics, of course, and people in bioinformatics for the treatment and modelization of the data that we will obtain. I will discuss that a little bit later on. So this is the way the samples are collected and processed. You must know that in the seawater, the plankton community's concentration is very dependent on the organism or the cell size. So you have a lot of viruses and very few metazoans if you take a constant amount of seawater. So for sampling, we use, in fact, progressive amount of seawater depending on what type of organism we are targeting. Up to 10 liters is sufficient to have a vision of the diversity of viruses. But if you want to go up to the large zooplankton, up to 2 millimeters approximately in Tara Oceans, you need to collect hundreds of thousands of liters. So this is not feasible by pumping, and we use nets to do that. So there are different instruments on board to collect the different size fraction that will be used to analyze later on either prokaryotes, small protists, large protists, or metazoans. So this is the back of the boat where most of the instruments were installed. You have the classical type of instrument to measure physicochemical properties. You have the nets to get metazoans, the pump here to pump water for prokaryotes and small protists, and we installed a small wet lab where filtration takes place. You have just 1 foot here, so this is the place where samples are filtrated, processed, and put in liquid nitrogen immediately. And there is also a dry lab dedicated to some imaging that can be done on board to look at the complexity and the nature of the organisms that we are seeing at the time of processing of one station. For example, this is what can be done using a flow cam for visualizing plankton on board. So these images are generated almost every day, and we record it, we stay with it. So this is all the type of different life forms that can be seen in a specific station. And from this data, sometimes we can take decision to continue or not to continue sampling, because this is more or less something that we already see, or this is something new. Here you have an image of many protist and forms that can be seen in one single station in the Mediterranean, which has been a bit redrawn. So you have all the forms that you are seeing here are single cells, and they are producing at a single place, and they are producing this very different architecture based on a type of exoskeleton on silicium or carbonate that are being used by these very peculiar organisms that we can see in the sea. So the ultimate goal of the project is really to combine all the genetic data, genomics and expression data in a metabolome mapping, and create with those data complex network of taxa presence, gene expression, and work later on in the next 2 to 3 years with people working on modeling ecosystem to have a global map of what type of plankton were present at the time of sampling of Terra oceans, and when, how, using oceanographic modeling, we can imagine that these communities will be changing and will be affected by the course of climate change, for example. Here you have, for example, physical measurements of particle abundance that we have looked at. This is a way to look at up to 500 meters depth particle sinking. So this is related with the carbon pump, the way that organisms after death are sinking. You can see what is already known, that in the North Ocean, in the Antarctic, you have a lot of activity of sinking, and there are many places in the tropics where this activity is extremely low. You have very, very few carbon fixation in these places. The same thing can be seen with oxygen, with many places that are more or less devoid at depths of oxygen, and these zones are indeed extending. So we sample specific environments using these measurements and choose the way we select stations based on this type of data. Okay, so let's go back to our subject, how to do eukaryote meta-omics in these communities, because we know how to do more or less prokaryote meta-omics, we know how to do viral meta-omics. Eukaryote meta-omics is really the challenge that is faced by this project in particular. So what we do in the frame of Terra Ocean is first to describe the communities with a phylogenetic marker, a part of the 18S gene. We tested different approaches for metagenomics, for metatranscriptomics based on poly-A selection, and single-cell isolation and sequencing. First, we want to have an idea of how complex these communities were and how different in complexity were the different size fractions that we were sampling. So this figure is the same as I shown before. It shows the different size fractions that were used for the sequencing process, so at least some of them. And here you have a barcode diversity, so the diversity of the part of the 18S that we were using for measuring eukaryotic diversity according to size fraction, from very small, here it's 0.8 micron to 5 micron, to large size fraction, 180 to 2 millimeters. That's what was done. And what you can see easily is that there is a decreasing in complexity with the increase in organism size. So it's not the same thing as a decrease in concentration. We really see that most of the complexity in the planktonic organisms, at least for the eukaryotes, are really present in the very tiny cell size fraction, where there is very little morphological knowledge and probably a very, very high level of genetic diversity. So this is a process of nucleic acids in the Terra Oceans project. I will not describe everything. I will greatly focus today on metagenomics. Just to know that we have these different components. We have also single cells that I will talk a little bit, and everything I will discuss today, I will not tell that anymore, is done on Illumina high-seq sequencing. So this is the first results we get on trying a very simple, basic approach on metagenomics using eukaryotic size fractions. So here you have size fractions that are greatly dominated by eukaryotes. They contain very few prokaryotes. And you assemble the data after sequencing one Illumina lane. We obtain contigs. So this means that it is possible, even with this highly complex community, to obtain contigs with the depth of sequencing that we can have today. And what we found is a very high number of genes that have absolutely no match in the databases. This was more or less expected, so it's what we show here. Here you have the NCBI content in terms of taxonomy, dominated, of course, by opisthocons and plants. And this is the 18S survey from Tara Ocean, part of the 18S survey, which is not that dominated. Of course, there is no plant here, but you can see that there are other type of animals and many protist lineages here. So when you go from the 18S to the metagenomes, you can see that these new organisms translate also in a very large number of new genes that have absolutely zero homology, even at very low stringency in the databases. There are other things that you can do with classical metagenomics, with sequencing and assembly and gene prediction. For example, after sequencing multiple samples, you can look at patterns of co-occurrence of genes, so co-occurrence of taxa. Here you have two different curves, one in blue, one in red. This one is not based on gene. In this case, it's based on molecular markers. And you can see that you have two different markers that are almost systematically co-segregating across a series of stations, which are here in the axis. So in these cases, we have patterns that are clearly suggesting that two different organisms are either loving the same environment or interacting by some way. And we can build networks from this data. So here is part of one of these networks that encompass viruses, eukaryote, archaea, bacteria. So this came really from metagenomics data. And you can see interactions. In green, you have positive interactions. In red, you have negative interactions. And you can see that using this type of networks, we have recently succeeded in looking at known interactions, and we rescue some of them. For example, here, you have interaction between a virus, which is called a coccolithovirus, and one cell, one coccolithophore, which is a single cell eukaryote. Here in this photo, you can see the viral particle attached to one cell. And this type of interaction is seen in our network. We can see that there is really a co-occurrence of this viral particle with some specific type of genes from these organisms. Okay, so looking at eukaryote genomes in metagenomes is feasible with at least a high coverage of short reads. Unfortunately, this is only part of what we can do, because only part of the information is going to the assembly. In general, we have 1 to 5% of the reads that we generate that enter into the assembly. The rest cannot be assembled at this coverage. We can augment the coverage, of course, but it will be not very cost effective at the end. So we try to develop another method to use directly the read information to look at ecological information in the different Tara Ocean stations. This method is called compare reads, so it has been published recently. Compare reads is not only dedicated to eukaryotes, it's dedicated to use very large amount of reads generated in metagenomic experiments to compare two different environments by comparing the content of reads. Very basically, we will define an intersection between two different environments by the number of reads that are common. And in order to have a reasonable time for calculating this interaction, we look at a method that looks at keywords inside reads and compare keywords between reads. So basically, when we say two reads are common, it means that they contain two different k-mers. So they are overlapping by, in this case, 66 bases in reads of 100 bases. The expression of the results of compare reads is basically a matrix, but it's a matrix bidirectional because you have a number of reads in one sample, number of reads in the other samples that are recognized one by the other. One read in a sample can recognize many reads in one other. And at the end, we obtain a large matrix. I will show you an example that can be used as a proxy to the genetic relatedness of two different environments. So we will use, as an example for one compare read experiment here, a system on which Tara Ocean had made a number of sampling. This is Africa here and Cape Town. So here we have a well-known system of contacts between the Indian Ocean and the South Atlantic, which is called the Agulhas ring system. You have a current that comes here through the Mozambique Channel, and it encounters here the Southern Ocean current, and there is a phenomenon called retroflexion. The current is going back, and at this point, there are creation of a number of eddies that in fact are ejected from the system and run through the South Atlantic Ocean. This is for one example of those seen from satellite. So it's very, very big structures. They can be hundreds of kilometers in diameters. And you can see just by satellite imaging that they are very different from the surrounding environments. Here you have South Atlantic waters, and here you have these eddies that came from Indian Ocean waters with very different characteristics, salinity, temperature, and so on. And these will cross the Atlantic Ocean up to the coast of Brazil and being progressively deformed and losing their speed and so on. But they last for a very, very long time. And this system is really interesting because it has not been a lot studied in terms of organism composition. Here you have a contact point between two different oceans, and these structures are able to take a community from one ocean and go through another ocean. And we don't know really what will happen with these communities. Will they be lost? Will they be part of the new system in the Southern Ocean? So in yellow here, you have the path of Tara across this system and the stations that have been sampled. And in this part of the project, we sampled two different structures. We call them rings. One young ring here, which has about 6 months, so it means that we know that its formation came 6 months before. And at the end, we sample a very old ring, a different ring, which has crossed the ocean, and it takes 3 years approximately to go up to the end. So we have also sampled stations here, so which are typical Indian Ocean stations, so the type of water that is being injected in the South Atlantic. So this is the results of the compare reads. Here you have a big matrix with all the stations and all the depths that we have sampled for this particular part of the project. And you can see that there are very expected results. For example, these few stations here are stations of the Southern Ocean. So Antarctic waters, we know that they are not a lot in contact with the rest of the system. They are really isolated. And you can see here that there is a lot of homology between the different stations close to the Antarctic, but there is almost no homology to all the other South Atlantic or Indian Ocean. So this is a good control for what we are doing. And here are the stations from the Indian Ocean. Here's the station from the South Atlantic, containing the rings. And you can see here and here the different results of the comparison between Indian Ocean and Southern Oceans that you can analyze to see in what situation you have more or less homology. So the way we did that at first is to look at intersection between the two stations that correspond to the large ring structure that I've shown you, station 68 and 78. We look at their intersection and the intersection of this intersection with each other station. So this means that we can see what happened to the very specific plankton community present in the rings, at least in the two rings that we have been able to sample, or in all other environments. So are the rings retaining a lot of the diversity present initially in the Indian Ocean? Are they progressively seen entering of the Southern Ocean plankton communities? And is there any type of exchange with the Southern Ocean structures? And some of the preliminary results are here, but they really confirm that we can use this type of approach to look at this exchange in diversity. What we see is that a large number of reads are common with the Indian Ocean stations, which means that the Indian Ocean community are really injected in the Southern Ocean, and they can last at least up to rings that are 3 years old. By contrast, if you look at South Atlantic stations, you have a decreasing amount of homology. So this means that probably in the Southern Ocean, those that are closer to Africa are more related to what is present inside the ring, probably by contact. And you see very different communities when you go toward the coast of South America. And as expected, you have only background homology to Antarctic waters. So this way of studying metagenomics, even if it's not looking for the moment at genes, it permits only to compare samples, but it's very useful for ecological purposes, and it will have extension because we have collection of reads that are homologous to different samples that can be further remapped when we created collection of interesting genomes or interesting genes that are really present in the plankton. So in resume for the metagenomics analysis that we have presented here, we can assemble significant fractions of eukaryote genomes, relatively small, but at least I think it's the first time that we see that we can really, in very different environments, obtain contigs of eukaryote metagenomes and use them to predict genes. And we can also use the same type of data to make read-level comparisons for studying in situ the plankton population relatedness between very different environments. So these results are interesting, but they are limited in scope. You see what we can do with that. And we would like to do more, in particular to have more genes predicted in the different environments. We would like also to have more reference genomes, because we are really lacking reference genomes that are ecologically relevant in terms of what is present in planktonic environments. So for this, we will use single-cell genomics and some metatranscriptomics. I will just show you very briefly what we can see in these first approaches. And this can be used to gain complementary insights from the metagenomics. So we don't see it as two completely different categories, but rather categories that we will try to mix. I will show you how we expect to do that, to obtain insights not only in the genomic composition, but also in gene expression, and also species divergence using reference genomes. So the effort to produce single cells from eukaryotic planktonic organisms is resumed here. This is a work done at Bigelow Laboratory, and the first stations that have been studied were in the Mediterranean and in the Indian Ocean, part of the complex that I've shown before. A little bit less than 1,000 different single cells of high quality were obtained, which meant that these single cells were amplified, they succeeded in amplification, and we were able to identify them using 18S sequencing. Here is a resume. We can separate phototrophic and heterotrophic cells in the flow cytometry process. So here you have the number of phototrophic cells and heterotrophic cells. All of them are single cell, of course. And here is a graph showing the diversity obtained in single cells compared to the diversity obtained in the same stations using the 18S survey. So the bars here are to be made in relation with the abundance of the organism. So this is the most highly abundant organism in the 18S survey. And here you have a decrease in abundance. This one is rather abundant, and so on. And here you have, by red point or green point, the single cells that have been obtained either using the heterotrophic sorting procedure or the phototrophic sorting procedure. So what can be seen here is that we have, in our single cell collection, we have a very large diversity of the different eukaryotic lineages that are present here, with lineages that contain sometimes both phototrophic and heterotrophic organisms. This is because they can appear in the two processes. And even if some of them are not present, for example, we have no ciliate, but ciliate are generally larger cells than what we can obtain in the process that we are using here. But we have a fairly large diversity that will be extremely helpful to create genome references that we are extremely lacking today to explain most of the diversity that we are seeing in metagenomics. And the last step that we are pursuing now is to use metatranscriptomics on the same samples. The same DNA, RNA extractions are subjected to metatranscriptomics. So we produce poly-A-based metatranscriptomes for tackling eukaryotic gene expression. We also produce the same thing for prokaryotes that I'm not showing here today, but we are producing poly-A-based metatranscriptomes. And these, for each sample, are used to map on references, either references for the few genomes that we already have today. The references can be the single cells that we are producing, or they can be a collection of genes that we are producing using the metatranscriptomic reads after assembly and a phase of clustering. So we have, in this way, we are in the process of constructing a very large construction of eukaryote-specific genes in the ocean. The first versions are tens of millions of contigs after clustering, so it's a very, very large collection that can be used to measure the activity and the presence of almost every gene in the collection based on mapping of meta-G or meta-T reads that we have obtained before. So this will lead to a normalized gene expression index for every gene in the collection, every gene in the reference genomes, based on the ratio between meta-T and meta-G, of course. Here is just an example for the last example I will show today on a particular organism, extremely abundant, an heterotrophic organism that is very, very highly prevalent in the oceans. It has just the name of mast 4 today, so it means marine straminopile number 4. There are many lineages of this type that have been sorted out. We know that this corresponds, in fact, to a number of species. There is not only one species here, right? It's a very generic names after recognition by 18S sequence homology alone, and we know that there are a lot of different species present here. One of them correspond now to a sequence genome, a sequence single-cell genome that we have obtained. Here you have a picture of what can be seen of this organism in fluorescence, so we can distinguish almost nothing because it's a very small, tiny one. It is part of this very, very small organisms, less than 5 microns, that contain most of the diversity I've shown you before in the planktonic eukaryotic organism. It's very simple, it's not photosynthetic, and it constitutes about, this organism constitute about 10%, a little bit less than 10% of all the marine heterotrophic flagellate. It is estimated as 10 to the 20 cells in the oceans. It's something extremely abundant, extremely unknown, and here with a crossing of the data we obtain basically using the single cell data, the gene prediction, and mapping the metatranscriptomic data. We can see here different stations and different depths, part of the Tara Ocean stations, and here is a profile of gene expression. In green, it's a gene that is discovered as expressed in this environment. So we can study now where this particular organism is present, where it is highly expressed, where it is not. In this very simple case, I didn't detail anything, but we can see that we have specific ecological features that appear each time with this particular organism. In particular, we detect transcripts here in some oceans and not other. In this case, it's the Indian Oceans, Pacific Oceans, where this organism seems particularly active, and it's present in surface water. We see it very, very at very low expression when we go a little bit deep to the other depths that have been sampled during the Tara Ocean expedition. So this is my last slide, so I finish here. The message is really that we are building a collection of genetic data based on meta-omics and different types of meta-omics that will be probably useful for many people working in planktonic systems. We plan to make things available shortly and to release data very regularly in the near future, to help people working on this organism access a collection of well-curated data sets for working on this. And to finish, I would like to thank people in my lab that works on this project, in particular for sequencing, and we are doing most of the sequencing and development of new sequencing technologies for adaptation to this particular project, and in bioinformatics, especially Olivier Jaillon and Eric Pelletier, who did a lot of the work I presented here. And thank you.
Thank you very much, Patrick, for a fascinating journey. So, are there questions for Patrick? Yeah.
Just curiosity, how do you balance life and job during the 3 years project on the ship? Thank you.
Sorry?
how do you balance personal life and job during the 3 years of the
What do you?
Balance your personal life and the job when you're 3 years. Maybe you should explain the annex for 3 years.
No, well, this was a well-organized expedition, and nobody spent a lot of time in the boat, in fact. People were relaying. People spent, in general, between 1 and 2 months in the boat. And then both the sailors and the scientists were replaced. It's a very exhaustive time of working there. Each station is about 48 hours of continuous sampling. And you can do that 3 or 4 times during 1 month, so it's very exhaustive. And no, we don't think that we could have done that for a very long time. So the organization was that there were a lot of scientists implicated. There were 15 people on board, 6 or 7 scientists, and in total, maybe 100 scientists have been on board during the cruise.
I'm sure there will be many candidates in the room to balance their personal life with stay on the boat. I would personally.
It's an interesting experience. Yeah.
So you showed that you discovered a lot of new organisms. I mean, did you do some phylogenetic analysis to see how much they relate to known organisms, and is it changing as well some taxonomy and some knowledge on eukaryotic?
Yes, well, there were some previous things that were known. We didn't discover everything completely new, but the 18S analysis in particular has revealed very new taxa, including what seems to be new phyla, because they were not homologous to anything known. It should be said that the depth of sampling, of sequencing sampling, is rather high. Each point was done with about 2 million Illumina reads, so it's something that has not been done at these depths across all these oceans. And the results are still being analyzed, but we think that the level of novelty that we are discovering here is very, very high. Maybe 50% of the taxons that are described by 18S were not present in the database before.
Oh, there is another question. Yep.
Did you study any of the experiments that used iron seeding on a large scale to see what is the impact using the same methods?
No. No, we didn't.
All right, so there are no further questions. I want to join me to thank again the 2 speakers of this morning.